Hello students, am I audible and visible? Let me know quickly if I'm audible and visible. Yes? Yes, we are live. Okay, 1 minute. Yes, is there any voice disturbance or anything? Yes, clearly audible and visible? Okay. Yes, fine. No issues, right? Okay, let's start the class. Let me tell my students, all sleepy people, I have to keep reminding. Okay. Yes, ready? Okay, students, so today we will be doing microbes in human welfare. It's a very small, cute chapter. And what is the best thing about this chapter is most of the students think that, oh, I will do this chapter one day before NEET. Okay, I will not do it anyways now because it's such a small chapter, and what could even come from this chapter, so I will just do it last minute. Okay, but let me tell you all this, from this chapter we can expect one question or two questions. That means 4 marks to 8 marks is guaranteed from this chapter, and that 4 marks and 8 marks you will directly get from this chapter. See students, now if I teach you biotechnology or if I teach you locomotion and movement, your marks guarantee is very, the comparison is very less. Like out of 10 questions, maybe you are able to solve only 8 of them. 2 questions gone. But in this chapter, it's not like that. It's like do or die. Okay. So we are going to do a do or die chapter. Okay. We have already, yes, we will complete this chapter today. Yes, Madhumita. So share the link quickly with your friends and let's start the class. So look here, like I told you, in previous years also one question was coming. This year we are expecting 2 questions because again, strategy chapter is removed. That's why we are expecting 2 questions. And today's session is going to be in such a way that I am going to help you decode NCERT, and we will write everything, we will underline whatever is necessary because everything is given in NCERT. There's no additional information given, and I will try to save as much as time, so I will do it quickly because I know that applications are out and now time is very precious for my NEET students, so I will not take any, not even a minute extra. We will just do it as quick as possible but in a thorough way. Ready? All of you are ready? So you know, if you are ready, what you need to do in the chat box. Come on. If you are ready, you should know what you need to do in the chat box. Come on. All of you. Then we will start. Yes? Let's see. Hello. Hi, all of you. Did I forget to wish you all? You will tell. Yes, it will definitely help you. And let me tell you, students, especially for the board students, I was checking your CBSE sample paper and 2 questions have come from this chapter. 2 questions, and both are 5-mark question, and I think one is a 4-mark question and one MCQ also has come. That means 10 marks question has come from this for boards. Do you realize? Yes, very good. All of you understood. Okay? Okay? Yes, Kaushal, your 10 marks will come from this chapter. Yes, okay. Students, we will start. Okay, so microbes. So when we talk about microbes, what do we know? We always think microbes always causes that some kind of a disease, right? Or it is of no use for me. But actually microbes are very, very helpful. So this chapter, see, all this while in NCERT they were telling, oh, microbes are bad, algae is like that, fungi is like that, virus is like this. So now they are like, okay, let's be in good terms with microbes because what if it affects me? So now they are going to talk fully good things about microbes in this chapter. Okay, so what you need to do is you need to remember, okay, right? Principles of inheritance I will do. Students, do not shoot me. Okay, I have been getting a lot of messages. I'm not able to sleep because I'm scared when you people will shoot me at sight because I'm not doing principles of inheritance. Wednesday I'm doing locomotion and movement, which is one of the most requested chapters. So like that, slowly I will do. Okay, slowly I will do. Okay. Hi Pravalika. Okay, so students, look here. Besides macroscopic plants and animals, microbes are a major component of the biological system on this earth. All of you know this. Okay. You would have studied diversity of living organisms in your smaller classes. Even in 8th standard, you would have studied a chapter called Microbes, a friend or a foe. Like, is it an enemy or a friend? Right? Do you remember which kingdom among the living organisms contain microorganism? All of you tell me in the chat box, which is the one? Which is the one where this microorganism will belong? Okay. So, which are the ones that are microscopic? So, basically, students, whenever I talk about microorganism, I have to remember this word. Microscopic, right? And it is not visible by the naked eyes, right? It is not at all visible by the naked eyes. Like, until I have a microscope, you'll be like, ma'am, if you ask me, like, did I see a microscope? No, sorry, if you ask me, did I see a microbe? You'll be like, I don't have a microscope. What are you talking about, right? Yes, that is true. Because you cannot see it in naked eyes, right? You cannot see it in naked eye. Oh, went off hero. Okay, microbes are present everywhere in soil, water, air, inside our bodies and that of the other animals and plants. So, microbes are everywhere. Okay, every single place microbes are there. Okay. Monera. Yes, Monera is the correct answer. Correct. Microorganisms, smaller. Actually, students, it was put into Protista, then it was brought back to Monera. So much confusions were happening, right? But if you see microorganism, it is there in fungi, Protista, Monera, all of this, right? Even fungi is there. Yes. And even virus is there, which is not put into any categories also. Okay. Now, they are present even at the sites where no other life forms are there. So, students, this line is very important. They can withstand, okay? They can withstand extreme climatic conditions. Did you all like the session? Did you all mark your attendance? How will I know how many of you attended the class? How many of you are watching recorded? Right? I need to know, no? Who are my regular live students? Who are my recorded students? So quickly like the session. Come on. Let's mark the attendance. I should see 42 likes. Come on, come on. Now it's 24. Ulta kar do, 42. Okay. So they can withstand extreme climatic conditions, right? So we know thermoacidophils, right? Methanogens. Then we have acidophils without just by, who can withstand acid. There are thermoacidophils. That means they can withstand acid as well as high temperature. So there are microbes everywhere where even human life cannot be there. Okay. Fine. Students, look here. Where the temperatures may be up to high as 100 degrees Celsius. Deep in the soil, under the layers of the snow, several meters thick and highly in acidic environment. Okay. Microbes are diverse protozoa. Okay. Bacteria, fungi, microscopic animals and plant viroids. Right. All this we have studied. Okay. That is what they're telling. So students, here they are, one second. Yes. Here they are talking about all the general things. Is the image visible? Yes, the image is visible. So students, look here. We have a set of images given. I have made everything into black and white to make sure that my students are able to see, because you will complain, ma'am, if you put white images, I cannot see. So that's why I made everything black and white. Please look at your NCERT. Colorful diagrams are there. Okay, students, this first one is rod-shaped bacteria. Everyone who has studied biological classification will know rod-shaped bacteria. Other name. What is the other name for rod-shaped bacteria? All of you quickly tell me in the chat box. Wow, I can be a rapper, no? All of you quickly tell me in the chat box. What is rod-shaped bacteria? Come on. Rod-shaped bacteria's other name. Okay? By then I'll label this. This is spherical. Spherical shaped. Right? And this one is a enlarged image of this rod-shaped bacteria. Very good, very good. Bacillus, right? Bacillus is the other name for rod-shaped bacteria. Okay, now spherical shaped bacteria. Why are the answers so slow? Quickly, the answers should be quick. Yes, Bacillus. Here it is. Coccus. Right? Student, this one's enlarged image is this one. Okay, this one. So what do you see here? You see a flagellated bacillus. Students, can all of you give me one example of bacillus? Very good. Coccus. Can you give me one example of bacillus? Quickly. Okay, this is the diagram. First one, rod-shaped bacteria, bacillus. Coccus or cocci. Okay, spherical shaped bacteria. here is an enlarged image of the bacillus where you will see flagella, which will help for their movement, right? Which will help for their movement. Yes. All of you give me an example. Very good. Example is lactobacillus. Okay. Give me an example for coccus streptococcus, right? We studied in human health, right? We studied in, what is that, molecular basis? Streptococcus, right? Pneumonia is there, many are there. Right? Streptococcus, pneumoniae, lactobacillus. Clear or not? Students, images are very, very important. That is why I'm doing images, because they might ask you to identify. They can give this for the exam and expect you to identify. So you cannot lose any marks, right? Yes. Students, look here. What do we have here? We have a bacteriophage. All my students who have done molecular basis, okay, in your life once at least, you will know bacteriophage or bacteriophage is a virus that can infect bacteria, right? It is a virus that infects bacteria. Yes or no? Okay. Then what do you see here? This is an adenovirus. Students, adenovirus is something that can affect the respiratory tract. Very important question can come. Which is the virus that can affect the respiratory tract? Okay. Yes. Yes or no? Clear? Yes or no? Yes, adenovirus. Correct. Okay, adenovirus. Yes, bacteriophage. Correct. So students, this is an adenovirus which can cause infection in the respiratory tract. That means difficulty in breathing. Students, all of you remember this word? SARS. During COVID time, this was one of the words that was highlighted, right? SARS. Okay. SARS is what? Severe acute respiratory syndrome. This adenovirus have the capability to cause SARS. Okay. Severe acute respiratory syndrome. Okay. Then students, if you look here, this is your tobacco mosaic virus. What is it? TMV. This has retrovirus and this has single-stranded RNA. Yes, this entire page is clear. Okay, entire page is clear. Yes or no? Simple, easy. Is there anything that is not visible? No, right? I think this is a little chill. Yes, clear? Okay. Yes, fine. Clear? Now next one. Students, look here. This is the next image. First, you will see a bacterial. So this I have done in my masters. Okay, it's a lot of fun. Okay, lot of fun. So students, what is happening? There, see, you can take any of the bacteria and then you take a Petri plate which will have some kind of agar medium which will actually help the growth of the bacteria. Then you take inoculum. Okay, inoculum, you tell that you dip it on the inoculum and you just rub it like this in the random pattern. So whichever pattern you rub, right, whichever pattern you go to and forth on the Petri plate, you will see the growth of the bacteria. So this is what bacterial growth. Okay, this is bacterial growth. This is what, students? Green color, right? Green color fungus. What is that? This is fungal colony. Yeah, fungal colony. Clear or not? So here we have bacterial growth. Here we have fungal colony. So fungal colony will be lightish green in color. Okay, lightish green in color. Okay, yes or no? Yes, streaking. Thank you. Ponyo. I was trying to remember what is that word. It's called streaking. Students, this is called as streaking, okay? There's actually different types of streaking. Now it's long time that I finished my MSc. I forgot all of this, okay? We used to do it in microbiology. So we have different types of streaking. So one type, like depending on which kind of bacteria, fungi, we have to do certain kind of streaking. Thank you so much, okay? Yes. This came in the victory test. Wow, Srinath, did you all answer? Very good. Very good. So students, images are important. That's why we are doing this. Okay. Yes. Now let's see what we talk about microbes. So students, I'm not going to read unnecessary things. I'm going to be sticking to the point. Okay. Yes. Students, a common example is the production of curd from milk. So now what happens? Now example, Sanvi is there. Okay, Sanvi is there, who is checking on my qualification, who is asking me to go to the previous slide. So many things is happening in his life or her life that they have forgotten to what their mother told. So mother had told last night, Sanvi, do one thing. Take 1 spoon of curd and add it into the boiled milk that I kept. Because Sanvi is so busy doing things in this life, he or she forgot. Now what will happen? Mom will scold in the morning. Why? Because students, actually that 1 spoon of curd which she asked Sanvi to add in the hot milk was an inoculum. What was it? It was an inoculum. So why was this inoculum added? Because in that 1 spoon of curd, who was there? Hero was there, right? Who was there? Hero was there. That is lactic acid bacteria or your lactobacillus was there, right? Lactic acid bacteria or your lactobacillus was there. So in this inoculum, lactobacillus was there that actually helped to convert this milk into curd. Yes or no? Milk into curd. Okay. Yes. Okay, fine. Clear or not? Yes, clear. Okay. So students, that is what happened. So what did we do? We took 1 spoon of curd, added into the milk, and that milk, entire milk got converted into the curd next day morning. Okay. So after that, what happened? Sanvi was out of the house. Okay. Now he'll be out of the live also. Right? Yes or no? Because he's not doing his job and he's checking on other people. Right? Yes or no? Fine. 1 minute. Yes, 1 second. Done. Okay. Yes. So lactobacillus grow in the milk and convert it to curd. So during the growth, okay, the lab produces acid that coagulates and partially digest the milk proteins. Okay, this is what is happening. So in the milk, right, in the milk, we added curd. And what did this lab do? Lab actually helped in coagulating the milk proteins. Okay, and converting milk to curd. Clear or not? Okay. Thank you so much, Kavya. No, drugs part is not completed. It will be done 1 day. 1 day. Let's wait for that 1 day to happen. It will be done soon. Yes. Now, a small amount of curd added to the fresh milk is called as inoculum. Very, very important word. Please write it right away. Okay. Inoculum or a starter. You can also call it a starter. Now, contain millions of lab. That is the reason, students. Your mothers will not know the scientific reason, but they know that if I add 1 spoon of curd into this boiled milk, definitely the milk will get converted to curd. Okay, which at suitable temperatures multiply. That's why usually it is kept at 42 degrees Celsius. So if you want, you can write it 42 degrees Celsius. Okay, but usually overnight we keep it and then aram se keep it in one side. Okay, yes. Yes, what is happening, yaar? Atiha. Atiha, I told you drug is not completed. Okay, it will be completed. Yes. Fine. Done? Okay. Now students, thus converting milk to curd, high Vijayan, which also improves its nutritional quality by increasing the vitamin B12. Okay. So students, actually, let me write it here. Okay, this curd No, this milk to curd. This curd has so many benefits. I am sure all of you know vitamin B12 is very high in curd. Okay, calcium levels are high in curd. It also helps in digestion. And nowadays people use it for face also. Right? People use it for face also. Right? Yes. So, vitamin B12, calcium, digestion. All this curd is very, very useful. So, you should tell when they ask you any kind of beneficial of why curd is used or something like that, you can tell it is rich in vitamin B12, it is rich in calcium, and it also helps in the gut health. Okay, it helps in the gut health. Is this clear? Is this clear? Can we move on? Right? Yes. Now, the dough which is used for making foods. Okay, students? Oh, 1 second. Board will come back. Okay, yes. Such as dosa and idli. Now, who is a South Indian in the class who does not know about dosa and idli? Come on, tell me. I think even North Indians love dosa and idli. Yes or no? Anyone in the class who does not like dosa and idli? Quickly, quickly. I'm waiting for your answers. Yes, very good. Yes. Why is milk first boiled? Milk is first boiled before, so that the milk does not turn bad. You know what happens if you keep milk outside and just go and sleep overnight? Milk will convert to paneer by itself. Okay, that means milk will get spoiled if it's not kept in refrigerator. That means the temperature is not maintained. So that's why we boil it to make sure that the milk does not go bad. That's all. Very simple answer. Students, so everyone like idli and dosa? Yes or no? Now you need to know idli and dosa. Luco Nostop is the person who actually helps in the puffing of the idli and dosa. That is why your mom grinds it the day before and keeps it in the fridge, and she makes it next day morning. She does not, the day she grinds it, she does not make dosa, right? That is for letting this dosa and idli batter to ferment. Most of the people write answer yeast as an answer. The moment they see dosa idli, they'll be like, yeast is the answer. Right? Yeast is not the answer. Leuconostoc is the answer, and it is not the nostoc that you studied in biological classification. That is different. Nitrogen fixation nostoc is different. Okay? Yes. Students, this is also fermented by bacteria. The puffed up appearance of the dough is due to the production of carbon dioxide. So this leuconostoc is going to produce carbon dioxide gas that allows that pores formation. Have you seen when you pour, right? Have you seen the small openings? Right? That small pore-like structure. That is because of the release of carbon dioxide. Clear? Now, students, can you tell which is the metabolic pathway in taking place resulting in the formation of carbon dioxide? What is the cycle? Okay. Now, where do you think the bacteria for this fermentation come from? Students, where will it come from? We neither add it, right? We add an external, or what do you tell? The rice, urd dal, everything mixes the batter, right? The batter itself will give that fluffiness, right? Keeping it overnight, automatically fermentation will happen. Okay? Yes. Similarly, the dough which is used for making bread, students, this is called as baker's yeast. How many of you have seen baker's yeast? If you go to shop and if you see, there will be a packet where it is written baker's yeast. That yeast is different from the other yeast that you get in the bottles. Okay, so this is baker's yeast which is used for making bread. Okay, now students, baker's yeast also called as Saccharomyces cerevisiae. So what is the baker's yeast name? Follow, repeat after me. Saccharomyces cerevisiae, baker's yeast. Okay, a number of traditional drinks. Students, toddy. I think it is also it's there in a lot of places. Students, how many of you have seen toddy? Okay, toddy is very common drink in Kerala. Okay, we call it kall. Okay, so there are places called as kall shop. No, not shop, shop. Okay, so kall shop, that means it is there in every nook and corner, students. And there they sell toddy. That means fresh toddy. They will not let it ferment for very longer time. right? They will not let it ferment for very longer time. So a number of traditional drinks and foods are also made by fermentation by microbes, like toddy, a traditional drink very common in Kerala and some parts of the southern India. I heard last time when I was taking class, I heard students telling in Andhra also it is very famous, okay, by fermenting sap. Students, so toddy, if you see generally, I think the high quality, the good quality one is in palm. That means palm wine, okay, it is from the palm tree. But in Kerala, most of the time I've seen it on the coconuts and the normal. I have very rarely seen it on a very, like, I think there is difference, right? Good quality, okay? Yes, they say it is healthy, but it is very khadwa. How do I tell it? Khadwa. Okay? Students, how do I tell it? It is very sour in taste. It's very sour in taste and it is not sweet. Kallu. Even same thing here. In Malayalam also it is called kallu. Okay? Yes. Okay? Not just because it's healthy, do not drink it. And it is present on the palm, type of palm called as Caryota urens is the complete name. Caryota urens. Okay? Caryota urens. So it is fermented sap and microbes are used to ferment fish, soya bean and bamboo. Okay? So many things with microbes, yeah. See, now microbes are... One second. Something is wrong today in this board. Yes. So microbes are also used in fermenting fish, soya and bamboo shoots to make food. Okay, make food. Now, cheese. So now what and all did we study? We studied milk to curd. Then we studied yeast. That is your baker's yeast. Then we studied... what is wrong? Dosa, idli. Okay, then we studied toddy. But in this, only two names you have studied. That is milk and yeast. Okay, I told you dosa, idli. It is not mentioned in your NCERT. So, you have to know lactobacillus. You have to know baker's yeast, also called as Saccharomyces cerevisiae. Okay. Now, you're going to see cheese. Cheese, very important. Cheese is one of the oldest food. Tom and Jerry, whoever has seen Tom and Jerry will know that there are different types of cheese, right? The cheese that you see in Tom and Jerry will be like this. Yeah, I'm an artist. Wait 1 second. Yes, this is the cheese that you see in Tom and Jerry. Yes or no, students? This is the cheese that you see in Tom and Jerry. The cheese that you see in home will be like this. Okay, that means a slice of cheese, then shredded cheese, then, you know, normal cottage cheese, which is used for very health conscious people. So cheese are of different types. Okay, now how can I tell the cheese is of different types? Students, first of all, do you see how many pores are here? So depending on the number of pores on the cheese, okay, the holes on the cheese, right, these are called holes. Okay, they can be different depending on their water retention. How much water do they hold? It can be different. Okay, depending on their flavoring, coloring, and from what it is made, it can be different. Depending on which kind of microbe is helping for the fermentation, it can be different. Yes or no? Right? Yes. Okay, fine. So students, look here. Different varieties of cheese are known by their characteristic texture, flavor, taste, and specificity coming from microbes used. Simple. Texture, water retention power, holes if they have more holes, okay, taste, and then depending on which microbes, okay, which microbes are affecting them. Now, look here students, Swiss cheese, first one. For example, the large holes in Swiss cheese are due to the production of large amount of carbon dioxide. So, these holes are because of release of carbon dioxide. That means more the carbon dioxide, more the holes, less the carbon dioxide, less the holes. Clear or not? Okay. Now, by a bacterium named as Propionium bacterium. Okay. Sharmani, full. Sharam is there, no? Sharam means what you know? Shy. Okay. So, who is shy? Propionium. Oh. Today board is dancing, students. Board does not. Okay, every cheese has different taste. 1 second, let the board little relax. Okay, then we will write. Yes, Propionium Sharmani. That means who is shy? Propionium bacterium. Sharmani, shy. Okay, Sharmani in Hindi is shy. Clear or not? people who don't know Hindi later will be like, Mom, I don't understand Hindi. Students, I cannot think of a word in our English or other languages. That's why. Okay, now we'll write all of this together. So I hope all of you have notes. I will just ask you to tell me. Now students, Roquefort cheese are ripened by growing a specific fungi on them, which gives them a particular flavor. Let's see if the name is there. No, I'll write it here. Okay, students, Roquefort cheese. Propionum Roquefort. Okay, Roquefort. Sharmani clear? Sharmani is clear, no? Yes, Propionum bacterium. Sharmani correct. Now we have one more person, no? Penicillium Rocky. Write it here. Penicillium. Okay, this is the name of the bacteria that actually makes Roquefort cheese. Okay, clear or not? Roquefort cheese. Okay, full, yes, full, what did I? English, all British born cheeses, all the names are like that. Penicillium Roquefort. Okay, now we will write all of this together. Do not worry, we will write all the household microbes together. Now, students, look here. Microbes, especially yeast. Now, my students will be fully interested because we are going to talk about alcohol, brewery, beer, vodka, all of this. Yes? Yes? Yes, thank you. Thank you, Atiha. Okay? Yes, students. So, how many of you interested? Okay, students, look here. So, from time immemorial, that means even from the time we did not know such things existed, these things were there. Who was there? Alcohol was there. Okay? So, production of beverages like wine, beer, whiskey, brandy, rum, all of these people were there. Now, how are they formed? Okay? So, for this purpose, the same yeast called Saccharomyces is going in there. Cerevisiae, okay, is used for bread making. No, the same yeast is used. Okay? Same yeast is used. Anyone who is interested in this, I will call your parents now. See, Baswaraj sir, correctly he came. Timing, you see, students, timing. Timing. Yes, okay. So, this is also called as brewer's yeast. Brewer's yeast. So, students, when they make these alcohol, that say, especially your beer and wine, it is a distillation, non-distillation process. And when they make the rest of it, that is your whiskey, brandy, gin, vodka, all of this, they do a distillation process. That means what is happening? Usually this yeast, that is your brewer's yeast, will kill itself if it makes 30%. Okay, more than 30% of alcohol. Okay, very controlling yeast it is. It will die when it makes 13% of alcohol. So now what happens? We cannot depend on this yeast for making the very concentrated alcohols, right? The alcohols like your whiskey, brandy, rum, all of this has like 40 to 60%, 40 to 70% of alcohol content. Now, how can I depend on this yeast who is going to commit suicide whenever it reaches 13% alcohol? I have to find another method. So, the other method they used was distillation. Okay, the other method they used was distillation. Clear or not? This much clear? Who is committing suicide? Why it's committing suicide? Be like yeast. 13% alcohol. Commit suicide. Yes? Okay, fine. Now, students, look here. Brewer's yeast is used for fermenting malted cereals and fruit juices. I'll tell you all of that to produce ethanol. Now, do you recollect? Okay, the metabolic reactions which results in the production of ethanol by yeast. Okay, do you recollect? Please recollect. Now, depending on the type of the raw material used for fermentation and the type of processing, two important things. Depending on the raw material and depending the type of processing. Yeah, come on, don't be depressed. With or without distillation. With or without distillation. Okay, let's write it now. All of you write with me. I'm erasing this. Okay, let's write some things. Now, students, beer, okay, wine, whiskey, vodka, gin, brandy, rum. Check if I missed something. Check if I missed any drink. Yes. So students, what is the benefit? No? Some of them drink beer and wine because of the concept that the alcohol content is less. Now what happens is, beer has complete sugar, wine also is completely sugar. So if you are planning to drink like, oh, there's no alcohol, so I'll drink 5 liters of beer, understand, you are consuming that much of sugar and you will put on weight. Okay, you will put on weight. So that is the thing with beer and wine. Okay, yes. Students, look here. This is beer is actually made of barley. Okay, and percentage can be between 3 to 6%. Okay, this is how much alcohol content is there. So whenever I'm writing the percentage, please write alcohol content, that is 3 to 6%. Okay, then we have wine. So wine is actually fruit juice. Okay, wine is fruit juice, but it is just sugar water. Okay, wine is fruit juice. Got it, all of you? Again, it is also somewhere between 9 to 12%. Sorry, 9 to 12%. Clear? Are you all there? Yeah, all of you sleeping? All of you sleeping? Yes or no? Sleeping. All sleeping beauties, wake up. I know it's a late class, so you will sleep. Huh? Okay. Students, whiskey is actually, let's take another color for our distilled one. Yes, it is actually fermented cereals. Fermented cereals. This will be 40 to 70% alcohol. That's why a person who drinks whiskey will suddenly be like, oh, the world has changed. I am the hero. I'm Shah Rukh Khan. I'm Vijay. Right? Suddenly he'll be like, oh, today I can do anything I want. They will go break here or there. Tomorrow they'll understand that, oh, it was whiskey who was talking in my head. I was not the person. Right? Right? I did not become any hero overnight. Nothing has happened. 40 to 60% alcohol went into my cerebellum and cerebellum lost balance. Yes or no? Yes? Okay, now brandy is actually fruit juice. Fruit juice fermented. Again, students, 40 to 50%. No, 40 to 60%. If I write less, no, they'll be like, oh, how can you insult us? Okay, all the people in the company. Okay. Okay, so clear? Even I sleep later than that actually. Yes? Yes, students. Now whiskey done. Vodka. Vodka is actually starch. Potato. So it is actually starch. Okay, so, and this is considered as a women's drink. This is considered as a women's drink. Okay, you know why? Because they say that if men drinks vodka, their sperm count will reduce. Okay, their sperm count will reduce. That is the reason vodka is told as a women's drink. Okay, this again has 60%. Okay, yes. Then did we miss something? Gin, I don't think gin, but anyway, just write it. Okay, it is made up of rye, malt, okay, wheat, again 60%. Yes, this is also again made up of starch, student. 40 to 70%. Clear or not? This much clear? That's all. That is your alcohol content. What it is fermented. Please remember, yeast will commit suicide, right? Yeast will die if the percentage go to 13% and above. Now what we have to do is, for these kind of alcohols, right? Your beer and wine, which I've written in green, understand, non-distillation method is used. That means, see, we can use the yeast itself. We can use our brewer's yeast because we are brewing beer and wine and both of the percentage are not crossing 13. Correct? But when it comes to whiskey, vodka, gin, brandy, rum, we cannot use yeast because yeast will not ferment. So that's why we use distillation method. Clear? Yes? Okay, done. I'll send you the notes, do not worry. Students, shall we quickly write all the household bacteria together? I mean microbes together. Yes or no? Yes? Fine. Okay. Yes, students. Let's write. Start. Okay. First one is your bacillus, that is lactobacillus. So we will write milk to curd. All of you tell me which is the one. Lactobacillus. Okay, lactobacillus. All of you quickly have to tell. Okay. Idli dosa, leuconostoc. Okay, which will actually help in fermentation. Yes or no? Yes. Next, what we learned? We studied bread, right? Bread. Bread is done by what, students? Saccharomyces. Always underline. Okay, always underline. Yeah? Yes. Next one. I'm using different colors, so it will be helpful for you. Milk, idli, bread done. Cheese. Cheese. Cheese, we have two types. Okay, one is Swiss cheese. What is the Swiss cheese, students? Propionium bacteria, no? Sorry. Bacterium Shermani. Okay. Then we have Roquefort cheese. Roquefort cheese is by Pencilia. Pencilia. Yes, you cannot hear, students. Yes, exactly. Okay. Yes, Propioni, Bacterium, Shermani, Roquefort cheeses, Penicillium. Yes, Penicillium, Roquefort. Clear? Done. Yes, very good. Okay, clear. Which color? Which color? Yellow. Still, what else did we learn? Toddy we learned, but we did not learn anything else about toddy. But still, we will write toddy. Type of palm is Caryota urens. Okay, Caryota urens is the type of palm. Okay. Then what did we study? Quickly, quickly. Chat has to move faster. We're doing revision. Dosa, idli, toddy, baker's yeast done. Yes, perfectly audible. Okay, fine. Yes, students. Then we studied last one. Beverages. So there again, same thing. Saccharomyces cerevisiae. Okay, students, this is also called as brewer's yeast. Yes? Okay, done. Yes. Something went into my eyes. Okay, fine, done. All the households are done. Check quickly. These are not households. These are, the last one is industrial, okay? But I've written all of the things which we finished now. Students, industrial, we have beverages, we also have antibiotics. We will see now. Okay, antibiotics, right? With and without distillation, we have clearly wrote it, so do not worry about it. That you can see in the previous slide. But did we revise everything? Quickly check in your notes also. I'll send the notes to you people, but still. Okay, fine. Now antibiotics. So antibiotics are what? Against something, right? Against something. Antibiotics is against an antigen. Antibiotics is against an allergen. Antibiotic is against any foreign particles, right? So students, antibiotics produced by microbes are regarded as one of the most significant discoveries. So actually, how did the discovery happen? So I'll tell you, wait 1 second. So in the 20th century and have greatly contributed towards the welfare of the human society. Anti is a Greek word. It's basically against life. Against whose life? Bacteria's life, right? Against bacteria's life. So anti meaning against, bio meaning life. So what does it mean, student? Against, okay, against life. Whose life? Bacteria's life. Yes. So how did this happen? So here, look here. Yes. Any doubts? Yes. Against antigen, correct. So, students, Alexander Fleming was a scientist. Okay. He was actually working. So, imagine you people also are working in a lab. So, you were working on a Petri plate. You grew some bacteria. Then you let it grow for some time. Again, it grew, so many grew. Suddenly, one day when he noticed that wherever this something is growing, that area bacteria is not growing. So he was very confused. He was like, what is happening? Then he understood. So initially he did not understand. Later he understood that it might be something that eats bacteria. It could be a fungus that eats bacteria. It could be something else, no, that is actually eating bacteria. So same way, students, this was his Petri plate. He was cultivating something. He was growing a bacteria. Then he kept it for some time for incubation, and then he noticed that something was growing there which was not allowing the bacteria to grow in that area. Then he realized that it led to the discovery of antibiotic. Yes or no? Clear? It led to the discovery of antibiotic. So which was the bacteria that he was growing? I don't think it's mentioned here. Here it is there, students. Story is here. I already told the story, but do not worry, I'll make you read the NCERT. Okay. Yes, okay, students, why am I telling studio? Okay, Alexander Fleming, while working on Staphylococci, that means what? This is a spherically shaped bacteria. When he was working on Staphylococci, that is what I showed you previously, he recognized that there was a region where bacteria was not growing. Okay, so Alexander Fleming with Chain and Florey, these two scientists also came together for the discovery of your penicillium. Discovery of your penicillium. Yes or no? Okay. Yes. Clear? Yes or no? Yes, fine. Okay. So, Penicillium notatum. Okay, Penicillium notatum was actually found by Alexander Fleming, like we tell, no, by fluke. By fluke this happened. So this is also by fluke, students. He was actually working on Staphylococci, which is a spherical bacteria in the lab. That time Chain and Florey came together. They were like, okay, let's work on it. And they came and found Penicillium notatum. However, its full potential as an effective antibiotic was established much later by Chain and Florey. Okay, understood? Yes or no? Chain and Florey actually helped in finding that. Okay, actually helped in finding that. Clear or not? Yes. Yes, fine. One second. God. Okay. Exactly. Exactly, Praveen, exactly. Okay, so this antibiotic was extensively used to treat American soldiers who wounded in the World War II. Okay, students, that's what I'm telling you. This was by fluke, but it actually helped during the World War II when there was a outbreak, like how we had COVID, no, like that we had an outbreak. Okay, when we had an outbreak of a disease, this penicillin notatum was very, very useful. Clear or not? Very simple. There's nothing to break your head. Just understand antibiotics. Also, microbes are the main heroes for the finding of antibiotics, and the main antibiotic found is penicillium notatum. Okay. Did we miss something? No, right? Okay, that's all. He washed, he grew, whatever happened, he's just talking about that. Now, students, after penicillium, other antibiotics were also purified from the other microbes. Okay, can you name some other antibiotics and find out their sources? Okay, so you can find out some more sources and write about it. Okay, antibiotics have greatly improved our capacity to treat deadly diseases, right? How many of you take a Dolo as soon as you feel a little sick? You feel like, oh, I have chills today, I have a slight fever. You'll be like, okay, I have to take a hotel Dolo. Yes or no? Dolo is not an antibiotic, but then still Dolo is taken like as if we are taking some everyday tea, coffee. It has become like that, right? So yes. So such as plague, whooping cough, then diphtheria, then leprosy, so many diseases are avoided because of the antibiotics. Students, usually no one reads so much. They will only write antibiotics, penicillium notatum, Alexander Fleming, Chain and Fleury. That's all. That's all is the keywords. Clear? Okay. Now, students, microbes are also used in commercial and industrial production. Very important. Even antibiotics are industrial because medicines, all commercial value. Even your breweries, that means your beer, wine, everything for industrial value. Again, you will also understand some more of them. Okay. Now, look here. Certain chemicals like organic acid. Students, so the production of organic acid was actually a very helped for the commercial value. Okay. I will send you one box which I have handwritten for you in the Telegram. I'll post it on the Telegram. That is Vedantu NEET English. So do join us. Okay. And do join our family. So there I will write a box completely. So you can just go through that box. That's the ultimate box. Okay. Yes. Students, look here. Example. Aspergillus niger, citric acid. Now this citric acid can be used for many things, right? Yes or no? How many of you know about citric acid? Okay, I will send you the box. Do not worry. Mainly citric acid is used as a preservative. Okay, preservative. Then we have acetic acid, vinegar. Okay, so yes, we have Acetobacter aceti, which will release acetic acid. Then we have Clostridium butylicum, again a bacterium which will release butyric acid, and Lactobacillus. Do I have to tell? Okay, so come all of you, write with me. Start. First thing, what? Aspergillus. Okay, Aspergillus niger. What will it release, students? Citric acid. And this citric acid will be used as a preservative. Are you all writing? Yes, are you all writing with me? Okay, good. Now, Acetobacter. Students, Acetobacter aceti. Okay, it is going to release what? Acetic acid, commonly called as vinegar. Okay, vinegar. It is again used in pickles. It is used in for making so many things, but I will just write it is vinegar. Okay, you can write uses. Mainly it is used in pickles and for cooking, right? Yes. Okay, very good. Fine. Students, then we have who? Clostridium. Clostridium. Yes, Clostridium butylicum. It will release butyric acid. There is actually a R. Okay, when we write butyric acid, we will forget the R. So do not forget the R. It is butyric acid. That is actually used for rancid butter. Okay, rancid butter. Yes or no? Rancid butter. Then lactobacillus, I'm not writing it here, but I hope all my students know lactobacillus, milk to curd. Yes or no? Yes. Yes, yes. Yes. Okay, done. Students, we have done the box also. Still, I'll send you the box. Oh, very nice. Yeah, done. All of you wrote? All of you wrote? Okay. Students, rancidity, you would have studied it in chemistry. I think in 10th you would have studied, no? The fat. I don't know, you should ask Diksha ma'am a lot about rancidity. But I remember learning it, rancidity, that means fat accumulation and then, I don't know, something about fat. Okay, that kind of that same rancidity, that same technique is used. That is called as your rancid butter. So do ask Diksha ma'am about this. It's a more of a chemistry word. Now if I explain, you will forget your chemistry. So yes. Students, now yeast is used for commercial production of ethanol. What is it used? Ethanol. Now where is ethanol used? In labs. Right? When you go to your chemistry labs and all, you will be using ethanol. Okay. Now microbes are used for the production of enzymes. Lipases, very important. Lipases is actually used for the detergent effect. Okay. 10th sample paper is there. Yes, rancidity. I remember teaching rancidity, but now I forgot. It's been very long. Right? Rancidity in 10th standard I have taught. Okay. It's something to do with fat and iron taste. Yes. Okay. So lipases are used in detergent formulation and are helpful in removing oily stains. So who is lipases going to help? Lipases is going to help to remove, okay, remove oil stains. Okay, students. So what is it going to do? You know, the formulation. So when you put soap on oil, what happens? That hydrophobic, hydrophilic tail, all of that, no, forms that micelle layer. Micelle layer. Again, this also you have studied in 10th standard. All if you remember this diagram in your NCERT, like this one circle will be there. Okay, like this one circle will be called. So same way the lipases also is going to do the help. That means it is used in the formulation of detergents. You must have noticed that bottled fruit juices brought from market are clear than compared to those made at home. Why? Why is the reason? This is because the bottled juices are clarified by the use of pectinases. That means, students, something that will digest pectin, protein, right? So, pectinases. From the term itself, do you understand proteins? So that is the reason why your Tropicano, why your Real Me, all these, will be so clarified. Like, Rasna, if you mix it in water, also clear juice. But at the same time, if you do it at home, have you ever seen, students, Appy Fizz and apple juice in your home? It will look like, it does not look like twins. It will look like born to two different mothers. Yes, we will not even feel like it is from apple also. Yes or no? Agreed, all of you? Yes. So this is pectinases which will digest protein. Digest protein. Clear? Okay. Now streptokinase produced by bacterium streptococcus is modified by genetic engineering, is used as a clot buster. So imagine you have a cardiac arrest because someone gave you a shock of your life, okay, by telling something or by doing something. So suddenly you have a clot in your heart. Normally it does not come like that. Okay. Yes, fresh juices is better than this one, student. That is just sugar water that you're drinking. Nothing is there. Protein also is not there. So suddenly you show, no, when your mom comes to scold you, you'll be like, I'm getting a cardiac arrest, I'm getting a heart attack. So other than that, the real people like so many of them have got heart attack, no, they will have clots in their heart. I have taught this in body fluids, okay? Remember arteriosclerosis and arteriosclerosis, right? So that time, they will give streptokinase. First injection, they will give a streptokinase for that person. So clot buster, okay? Yes. It is used clot buster for removing clots from the blood vessels of the patient who have undergone myocardial Infection leading to heart attack. Clear or not? Okay. No, we don't drink protein shakes. We only drink Appy Fizz. Okay. Yes. Done. Done. Another bioactive molecule. Students, cyclosporin A. All of you tell me, why is cyclosporin A used? Why is cyclosporin A used? Yes? Yes, Baswa sir is disturbing my class. Yes or no? Baswa sir is disturbing my class. Students, why are the likes so less? Yeah, not even 100. So sad. Not even 100. Okay? Yes. Too much gas and Happy Fizz. Yes? Clear? Oh my god, what did I do? Sorry. Okay. Exactly. Sir is also spammer. Okay. So students, what is happening? Cyclosporin A is an immunosuppressant, right? Immunosuppressant. Correct or not? Okay. Yes. Students, so before the body should not reject. Fine. Yes, very good, Madhumita. Yes, cyclosporin A is an immunosuppressant. That means when we are taking organs, like imagine you sell your kidney for buying iPhone. So after that, you're like, I want kidney from, you sell someone's kidney for buying iPhone. Okay, and then you have to return that kidney, or you want to give a kidney from somewhere. That time the body should not reject. No, that is for that you will inject cyclosporin A. Okay, you will inject cyclosporin A. Then students, look here. That is used as an immunosuppressing agent in organ transplant. Is produced by whom? Trichoderma polysporum. This question was asked in one of the years. Okay, Trichoderma polysporum. Made what? Cyclosporin A. Students, this year's CBSE sample paper 2023-24 also, it is there. Okay? Yes. NEET student, I hope you are a NEET student and can read the channel name. It is Vedantu NEET English. Okay? Okay? No, I didn't sell the kidney yet. But one day will come. One day will come. When we all sell. Okay? When they make 15 to 27 and then put 4 cameras and then, right? Yeah. So that time we will all sell kidney, students. Okay. Trichoderma polysporum is cyclosporin A, releases cyclosporin A. Okay, now statins produced by yeast, that is Monascus purpuris. What is it? Monascus purpuris. Okay, what does it release? Tannins. Okay, it will release tannins, which has been commercially used as a blood cholesterol lowering agent. Yes, blood cholesterol lowering agent. Okay, yes or no? Button phone is fine. Okay, fine. Good job, students. All of you have so much. What to tell? You love your kidney more than phone. Very good. Okay. Yes. Monascus purpuris is tannin, which is actually helping blood cholesterol lowering. Students, actually, that is the reason why men get heart attack more than women. The percentage, if you see, that is because in our women body we have estrogen, which is a natural blood cholesterol lowering agent. That is not there in men. That is the reason whatever they consume, the cholesterol will get settled down in the heart. Okay, yes or no? Very good. Okay, so it acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. Okay, yes, fine. Now, we know that large quantities of wastewater. Now, whom are we talking about? We are going to understand sewage treatment. Okay, I will teach you sewage treatment directly without reading your NCERT. Okay? Yes or no? Clear? Yes. Clear, clear. Okay, fine. Students, so sewage water, what is happening? The municipal wastewater is called sewage. So students, anything, be it organic matter, be it inorganic matter, be it the papers that you threw, the cigarette that someone threw, the lace packet that you had yesterday and you happened to throw, all this has to be filtered. So this sewage water will be filtered and then only it will be allowed to be, disposed into the river or lakes to avoid any kind of contamination or water pollution, and then this water can go to a tertiary level of cleaning to make sure that this water can be used for drinking also. Okay, yes, drinking also. So students, shall we start sewage treatment? Yes, ready, we should be able to complete this chapter fast. Okay, fine. So students, sewage treatment, if you start with, first what is going to happen, simple things have been done. Okay, 1 minute. Let me draw my art and the artist. Okay. Okay, sewage treatment means what? From where is it coming? It's wastewater. Yes or no? So we will just write the general thing. Okay, it is waste water treatment. So it is also called as sewage treatment plant, right? Sewage treatment plant, STP. Okay, STP. Sewage treatment plant, how does it happen? It can happen in two ways. Okay, so STP, I'll write off sewage treatment. So it's very easy, nothing to break your head. Okay, can happen two ways. Can anyone tell me which are the two ways? Students, physical, yes, 110 likes, very good. Okay, so it can be physical or you can tell primary, secondary. We will tell physical and biological. Physical, biological, or in other words, primary. Okay, primary or secondary. Yes or no? Primary or secondary. Now, students, physical or primary, that means filtering it other ways, like normal plastic cover, all of this. Very good. Lot of you told the answers. So now let's see. So the first way that government did was to make sure that we make sedimentation tanks, and also we make mesh, or you know how you have I don't know what do you call that, students, sieve. We call sieve, no, for chai. After pouring tea and all, we get that tea powder, no, called sieve. We tell sieve. I don't know what you tell in your own languages. So like that, they thought they will set it up. Clear or not? Yes, very good. Primary, secondary, you can tell. Physical, biological, you can tell. No problem. Okay. That is called as sequential filtration. So under primary, first one that you're going to study is sequential filtration. That means in a sequence, sequential filtration. Friends, that means in a sequence, we filter it. Okay, now imagine there is water, okay, lot of water, and then there is, say, chocolate packets. Okay, chocolate packets are there, and then maybe some other tissue is there. All this is there. Okay, now in the first step, what are we going to do? We are going to use a mesh, like a sieve, and filter this out. Now, what and all will get stuck? Imagine tissue, everything, it will get stuck. Okay, now when it comes down, what we will have? We will have water. Okay, we'll have the water. Imagine, students, you put a stick, like a ice cream stick. Ice cream stick is too small to go through the first sieve, right? So here the ice cream stick also will come. Okay, yes or no? Tissue, plastic cover, everything went. Now next to see what is going to happen. The size of the mesh, will reduce. Okay, size of the mesh will reduce. Clear? Sequential filtration. Okay, sequential filtration. Now what will happen? This stick wrappers also be filtered. Okay, this stick will get clot or caught here. Now only the water will come. Now who will come? Water will come. Is this clear to all of you? Sequential filtration, yes or no? Please tell me in the chat box. Okay, sequential filtration. Now students, when they saw, no, after some time when they're seeing that there are some floating debris. Now we physically removed all the big things which was there. Now they're seeing that there are some floating debris. Okay, very small things. What do? Floating debris. Now they were like, okay, sequential filtration alone will not help. We need to do something more. So here I am writing sequential filtration. Okay. Second thing they did was sedimentation. What did they do? Sedimentation. Okay. So from here, students, we took the water. We took a sedimentation tank. Okay. Okay, sedimentation tank. Now what is going to happen inside this? We will have, yeah, color keeps going. Okay. We will have water. Now this floating debris would have settled down, right? They would have settled down to the bottom. Okay. They settle down to the bottom, and that is called as, can anyone tell me in the chat box what is this? Sedimentation. This is sedimentation. Yes, correct. Sedimentation is the method. Now what is this, students? Floating debris, everything got settled down. Can I call it primary sludge? Yes, very good. I will call this primary sludge. Okay, I will call this primary sludge. Clear? So first we did sequential filtration. Then what did we do? We kept making meshes of different size. So even if there is a larger object, even there is a smaller object, all this will get filtered out. Then we notice there are some debris, floating debris, which cannot be taken by the sieve. So what did we do? We went to the second step, which is called as sedimentation. And in sedimentation, we let this entire water, which we filtered out from sequential filtration, to settle there, wait there. We just waited. Okay, after waiting for some time, we saw something got settled in the bottom and we named it as primary sludge. Clear? Okay, primary sludge. Yes. Now students, this water, no, which I have sedimented and got, that means most of the unnecessary matters have gone. Now what do I have in this? There could be fecal matter because it is sewage, right? Sewage water means what? It's coming from our households. Fecal matter would be mixed. So many other organic matter would be mixed. Now what to do? Now we have to take this for our we have to take it for our biological filtration. So this one, okay, this is your primary effluent, right? This will become your primary effluent. Now this will go down. Okay, this will go for a second step, that is your biological filtration or biological method. Okay, biological method or secondary method. Now what will happen, students? We are going to take an aeration tank. Not aviation, aeration. It will give me heart attack. Oh, what happened? Students, 1 second. Something board is in depression. Yes. Work. No. Oh, thank God. Came, students. Okay. Yes. Board is also giving me a heart attack. Yes. Students, we are going to take one tank. Yeah. 1 second. Work. Students, this is an aeration tank. What tank? Aeration tank. Okay, we label it. Okay, aeration tank. Okay, now to this aeration tank, I am going to continuously pump air. I'm going to continuously pump air, and I'm also continuously going to do mechanical stirring. Okay, mechanically mixing is continuously going to happen. Yes or no? Got it? So mechanical mixing also will happen. Now in this aeration tank, I have my primary effluent. Okay, I have my primary effluent. Now after some time, I will see something called as your useful bacteria. Okay, so first let's draw some water. Yes, we drew some water, students. Then we saw now we saw some flocks. What I call it? Flocks. So flocks are actually a group of bacteria or fungi which is actually going to is useful to break down the organic matter, because I was able to physically remove it, I was able to maybe settle it down, but I'm not able to remove the fecal matter. I'm not able to remove the organic matter mixed with water. So for that, I'm going to use flock, which is a group of bacteria or fungi which is actually going to help. Students, because I'm continuously pumping air, don't you think these flocks are aerobic bacteria? All of you tell me in the chat box. Don't you think the flocks are aerobic bacteria? Yes or no? Yes, very good. They are aerobic. Okay, they are aerobic microbes. That means they want oxygen for respiration or for their survival. Now, students, what is going to happen? As these flocks are there, or as these bacteria are there, BOD, very, very important term. Most of the students get confused. BOD, that means biochemical oxidation, biochemical oxygen demand. That means how much this aerobic bacteria is going to consume. Okay, aerobic bacteria is going to consume. So BOD will increase. BOD will keep increasing. Okay, so students understand how the relation is. If organic matter, okay, organic matter is more, bacteria will be more. Yes or no? If bacteria is more, the demand for BOD will be more. If BOD is more, that means dissolved oxygen, the amount of dissolved oxygen in the water is going to reduce. Okay, so dissolved oxygen will actually identify the purity. Okay, so what is happening, students? If organic matter is more, that means bacteria will be more. The amount of oxygen that the bacteria consume, that is your biochemical oxygen demand, will be more. If biochemical oxygen demand is more, that means purity of water is not there. It is more polluted. DO will be more, less. Yes? Yes? Yes or no? Tell me yes or no. Biochemical oxygen demand will be more. Dissolved oxygen. This dissolved oxygen is the oxygen that is taken in by your fishes, plants that grow, right? This is the good one. So if DO is more, that means what? The water is not that contaminated. But if DO is less, that means the water is not purified or it is mixed with organic matter. Clear? Any doubts? Okay. Yes, if more the bacteria, BOD, the demand will be more. But then BOD, that means what is going to happen? Your BOD will keep the demand will be more. Okay, demand will be more. Okay, so now we have to, okay, I hope this is clear. I'm erasing this. Now, look here, all of you. Now this one, we have to send it for purification again, right? This has to go for purification again. So for that to happen, what are we going to do? This flocks will digest all the organic matter in this. That much work is done. Okay, that much work is done. Yes, correct. BOD is indirectly means the measure of amount of microbes that are there. More the microbes, more the BOD. That means the demand is more. Okay, so that time DO will keep reducing. Dissolved oxygen for other, aquatic. That is the reason, students, when an entire lake is filled with other matters, plant, other thing, organic matter, of if people are, you know, if people are misusing the lake, okay, for other things, then you will see that all the fishes will die. That is because there is no dissolved oxygen underground. Okay, students, what is going to happen in this aeration? After this, flocks will consume whatever organic matter. Yeah. One second. Yes. Okay, what is happening? Here, look here, all of you. Students, before the board gives hand, let's finish the chapter. Okay. Now this one is, can I call this my secondary effluent? Can I call it my secondary effluent? Yes. The flocks have consumed all the organic matter. Can I call it secondary effluent? Yes, I can call it secondary effluent. Now the flocks which are there, right? That flocks, out of that, some will be activated sludge. Okay, it is called as activated sludge. Okay. I will put them into. So now this flocks, I will write it here only. This flocks will be, some of the flocks or majority of the flocks will be sent to anaerobic. Okay, anaerobic sludge digester. Sludge digester. So this aerobic bacteria will be consumed by whom? Anaerobic sludge digester. Some of the flocks I am going to send it back to the aeration tank, which is undergoing this process. Which is undergoing that process. Clear? Okay. So some flocks will be sent to this aeration tank. Again, next round of water will be coming, no? For that, some of the flocks will go to that aeration tank. Most of them will be activated sludge. Okay, this will be called as activated sludge. That will be sent to your anaerobic sludge digester. Clear or not? Okay. Yes, inoculum. We call it inoculum. This one which goes to the aeration tank is called as inoculum. Inoculum. So much clear, students? Any doubts so far? Please let me know. Otherwise, I'll continue. So, students, in the anaerobic sludge digester, two gases will be released. Carbon dioxide and your CH4, methane. Okay, methane. Now, this methane and carbon dioxide will be used for biogas production. Biogas production. Clear or not? It will be used for biogas production. All of you clear? Or is it only for two of you it is clear? All of you clear? Please let me know. Yes? Yes. Clear, all of you? Okay, students, so what happens, this secondary effluent is kind of now, you know, organic matters are not there, big lace packets, other things are not there. So I can send this secondary effluent to rivers and lakes, right? To rivers and lakes, making sure that there is no pollution. Making sure that there is no pollution. But imagine I want to take this secondary effluent for drinking purpose. Okay, I want to take this for drinking purpose. Then what will I do? Have you ever seen in your, I don't know, your water, RO waters? You will see UV, RO, that means reverse osmosis, O3. All this you would have seen on the drinking water that you're drinking, the machine. You would have seen all these three things. So students, your secondary effluent can be used for drinking once it goes through these processes. Neither passing UV rays or reverse osmosis or by using O3, ozone, no? This is called as your tertiary treatment. So this is only done when it is required. Okay, when it is required. Okay, this is tertiary. After this, if you want to do, you can do tertiary treatment. Done? Yes. Okay. Okay, fine. Then, that's why I've put an extra slide. Students, one more thing. I'll write it here only backside. Ganga and Yamuna. Okay, Ganga and Yamuna is there now. What is it called? Ganga Action Plan and Yamuna Action Plan. Why did the government do this? Ganga Action Plan and Yamuna Action Plan. Since both of this was done to protect and preserve these rivers. These was the initiative taken by the government because of the overusage. That means students, even for some kind of rituals, they were misusing Ganga and Yamuna. If all of you know, in Hinduism, we go to Ganga and when a person dies, right, the ashes, the banana leaf with whatever puja materials, all this we used to throw it in the Ganga river or the Yamuna river, okay? Even for other things also, people used to go there. Now what is happening, when all this is not filtered and it is kept for longer and longer time, the organic matter content increases in the river. The fishes, plants which was living underground, right, which we don't see also, they all started dying. Okay, and we could see the water, it was turning black in color. Okay, so that is the reason, to protect and preserve these rivers, they started Ganga Action Plan and Yamuna Action Plan, where they took the help of some NGOs to actually help them. Okay. Yes, fine. Now students, biogas. So biogas, like I told you, the anaerobic sludge digester will digest the flocks. It will release two gases, carbon dioxide and methane. This two carbon dioxide and methane can be used for the formation of biogas. Yes. Okay. So look here, in village side there is gobar, that means cow dung. That cow dung will be packed, dried. That will be used for biogas. Clear? Okay, so biogas is a mixture of gases containing predominantly methane produced by microbial activity and which may be used as fuel. So students, this is used actually in villages for cooking. Okay, for cooking. Now you have learned that microbes produces different types of gaseous end products during growth and metabolism. Okay. Now the type of gas produced depends upon the microbes. Very important. Depending on the microbes only, the type of gas also will be there. And organic substrate they utilize this. In example, so they're telling about cheese, all that. Okay, that's not needed. However, certain bacteria which grow anaerobically on cellulosic material. That means students, plant waste. All of you have seen plant waste. Yes, have you seen your grandparents taking this, tomato peel, onion peel, potatoes peel and putting near the plant, telling that this is a very good, like a vermicompost, right? It is a very good fertilizer for the plant. Same way, these people dig a hole, right? Dig a pit, compost, we tell. In that, we add all the fruit, vegetable, all the domestic waste into that, and we will let the microbes do the job. Right? We will let the microbes do the job. That is called as cellulosic material. That means plant material, no? Plant material. Okay, produces a large amount of methane along with carbon dioxide and H2. These bacteria collectively called as methanogens. Okay, this we have studied. We will study this in biological classification, and such common bacteria is called methanobacterium. So students, methanogens and methanobacterium. Yes, kitchen waste. Okay, methanobacterium, methanogens. Clear? Now, these bacteria are commonly found in anaerobic sludge digester. I told you this will release carbon dioxide and methane. Now, these bacteria are also present in the rumen. Whose rumen? Cow. Cow? Yes. So, in the rumen of the cattle, and a lot of cellulosic material present in the food of the cattle is digested because this is present in the rumen. Who? Methanogens are present in the rumen. That's the reason why we cannot we cannot just start eating like how cow and all eats. No, we cannot start doing grazing. That's because in our body, we do not have the enzyme to break down the cellulose. We do not have any kind of microbes or any kind of enzyme to break down complex cellulose. Okay, cellulose. That's the reason animals are able to eat it. Now, bacteria help in breakdown of cellulose and play an important role in the nutrition of the cattle. Okay. Now, human beings, they're asking the same question. Dung can be used for the generation of biogas. Okay, I will show you the image. Okay, now, so students, biogas we will study very simple way. Okay, simple way. Now, if you mix, that is your gobar, that means your cow dung, with water. With what? Water. It is called as slurry. Slurry. This was first found by your that is your Agricultural Research Institute and also your KVIC. I don't know how many of you know KVIC, that is your Khadi Village Industrial Organization. These two people were the first people to actually find biogas and to utilize, because students that time, see, nowadays villages have very less cattle, but those days cattle was the main medium, right? So they used to be like, why to waste all this cow dung, right? Even though they used to put some amount on the plants and they used to use for putting that rangoli and all that, but still they were like, we can make something massive out of it. That is when they thought about this biogas. So what do? They take gobar, mix it with water and make a slurry. Okay, that's what here is told. So what will they do, students? Which keeps on rising. So I will show you the digester. You will understand that. Now this slurry will be put or will be added into your biogas plant. Okay, I will show you the plant also. It is there in your NCERT. Okay, biogas plant. Simple thing, students, nothing complicated. Gobar plus water will form slurry. That slurry will be added into the biogas plant. Now what do you expect from it? What do you expect from it? To form biogas, right? So it will release biogas. And the waste from this, right? Waste from this will be used as manure. Waste from this will be used as manure. Clear, all of you? Simple. Yes, KVIC. Okay. KVIC. Khadi and Village Industry Commission. Any doubts? Any doubts? Please let me know. Okay. So yes, students, all of you understood this, no? So listen here. Look at this tank. Where is this? Yeah. Where did my tank go? Students, look at it. Okay. So you will have your gobar plus your water. All of this, the slurry, right? See here you can see dunk water. That is your gobar plus water. That will go into your digester. From there, what will happen? This cap-like structure which you see, that will keep moving up and down. It will keep moving up and down. This is called the gas holder. So inside this, who and all will be there? Methane will be there, carbon dioxide will be there, hydrogen will be there. All these gases will be filled into this. That will form the biogas, right? That will go up and go out as biogas. Now, gas holder. Now, what will happen? The waste materials, the whatever unnecessary is there, that will be used as manure. Okay, that will be used as manure. Done. Okay. Yes, that's all. Now, biocontrol. Students, this is almost ending. Our topic is almost ending. Biocontrol means, students, there is a lot of ways that I can control something. I can use harsh ways. I can use soft methods. Right? 2 ways for everything. Same way, biocontrol means if I want to control any kind of pesticides, weedicides, mosquitoes, any of this, I can either directly use a very complex pesticide or a weedicide. But if I have decided that, okay, let nature do the job, I will use some kind of a nature, like which will not harm the nature, at the same time protect from this. It is not killing it, it is controlling it. Okay, so that is called as biocontrol. So biocontrol refers to the use of biological methods for controlling plant diseases and pests. Okay, in modern society, these problems have been tackled increasingly by biotechnology method. All of you know. Okay, biotechnology method. Increasing by the use of chemicals, okay, by the use of insecticides and pesticides, right? Now, these chemicals are toxic and are extremely harmful. Students, now what happens is, you'll be like, what is the big deal? I'm just putting a pesticide, I'm just putting a weedicide. Now, this pesticide and weedicide are made up of very complex substances. Now, these complex substances will not get broken down easily. They will remain in the soil for years to come. Now, every time you grow a plant, this chemical will also be getting into the plant. We will be consuming it. Okay, right? Yes. So, students, extremely harmful to human beings and animals alike, and have been polluting our environment, soil, ground, water, fruits, vegetables. This is what I was talking about. Okay, this is what I was talking about. Our soil is also polluted through our use of weedicides to remove weeds. Now, biological control of pest and disease. So, students, here, okay, I will not read all this unnecessary things. Okay, here, look here. So what are they talking about basically in this paragraph? A key belief of whom? Of the farmers is that we should eradicate whatever is there in nature with nature itself. Okay, so that is the use of this. So therefore, works to create a system where the insects that are sometimes called pests are not eradicated but instead controlled. There's a difference between control and eradicate, right? Control and eradicate. Now, contrary to the conventional farming practices which often use chemical methods to kill both useful and harmful lives. Okay, harmful lives indiscriminately. This is a holistic approach. Students, again, story writing is there. Full NCERT story has been written. So please try to understand. Biocontrol means it is a method by which we control, okay, we control pest and weeds, but not completely eradicate them, like how weedicides are there and all that. Okay, yes. Okay, here they're telling how do we control? By actually by disturbing their life cycle, by disturbing, you know, generation after generation, right? By laying eggs, all that kind of disturbance if you do, we can definitely control them. Okay, clear or not? Yes. Now, they are very familiar. Students, here you have to know ladybird will actually reduce aphids. Okay, dragonflies will actually reduce the mosquitoes. Same way your trichoderma. Okay, I will read it for you, don't worry. So students, look here. The ladybird and dragonflies are useful to get rid of aphids and mosquitoes. So ladybird for aphids, dragonfly for mosquito. Please be careful. Okay, please be careful. Now, an example of microbial biocontrol agents that can be introduced in order to control butterflies, caterpillars is the bacteria Bacillus thuringiensis, your BT. All of you remember, we'll be studying this in your biotechnology. Okay, we will be studying this in your biotechnology. Yes or no? Got it? Fine. Okay. Yes. Now, these are available in sachets and dry form. So students, it is to make it easier for all the farmers. We just give them. Okay. We just give them. Clear or not? Yes. BT cotton. Correct. BT cotton. Okay. Yes. Fine. Students, 1 minute. 1 minute. Yes, clear. Okay. So students, BT cotton, this will be available in sachets. Now what they will do, we'll have to mix it in water and just pour it everywhere. That's all. Okay, very easily available. So it will help to, you know, control the larvae. It will go into the gut of the larvae. Look here, very important. It will go into the gut of the larvae and the toxin is released and the larvae gets killed. Okay, larvae gets killed. So this is how BT cotton works. So when a larvae consumes it, so what will happen? It will go into the stomach and kill the larvae. Okay. Now the bacterial disease will kill the caterpillars, but leave other insects unharmed. So that is the best thing. Genetic engineering, right? Genetic engineering actually helps in specificity. Specificity. That means what, students? If I am trying to talk to Madhumita, Kirti or YS will not come into the call. Why? Because it's very specific that I'm calling Madhumita, so I would only talk to Madhumita. Right? Same way, this genetic engineering is a technique where we have modified things in such a way that whatever is needed only will be there. Whatever is not needed will not come. Clear? Understood? Specificity is there. Okay? Yes. So because of the development of methods of genetic engineering, in last decades, the scientists have introduced that is your Bacillus thuringiensis toxin genes into the plant. Such plants are resistant to attack by insect pest. Okay, that one example is your BT cotton. Is one such example. Clear? Students, cotton is very, very difficult to cultivate. Very, very difficult to cultivate. That it can be affected very easily by any kind of pest. So when BT cotton came, no, it was such a relaxing thing for all the people who was cultivating cotton. Okay, because cotton falls off very easily. If there is a slight wind, cotton will fall off, then that cotton cannot be used. Okay, so there were like 100 problems. So that's why BT cotton was a savior. Okay. A biological control being developed to use the treatment of plant disease in is the fungus. Students, very important, Trichoderma. This Trichoderma species are free living, okay, and very common in the root ecosystem. They are effective biocontrol agents for several plant pathogens. So this fungus which grows, okay, this fungus which grows actually helps avoid other pathogens to grow on the root and not affect the plant. So Trichoderma is one such kind of a fungus which is free living, but it is actually going to help other pathogens to not affect the plant. Okay, so there are funguses which are good. Okay, then you have baculovirus, are pathogens that attack insects and other arthropods. Okay, baculovirus will attack insects and other arthropods. So if you are feeling that you did a farming and there is what to tell arthropods are affecting, okay, cockroaches are there or rodents are there or some other kind of butterfly insects also have come, that time you will have to use baculovirus. Clear? Specificity. Okay, now the majority of baculovirus used as biological control agents are in genes. Okay, nucleopolyhedrovirus. Okay, so why am I gibberish is becoming? Okay, nucleopolyhedrovirus. These viruses are excellent candidates for species-specific narrow-spectrum insecticidal application. Students, very, very important. They can ask you a question, which is the one that has a narrow spectrum, which is very, very specific. Okay. Now, they have been shown to have no negative impacts on the plants, mammals, birds, fishes, or even on non-target insects. Imagine by mistake an insect happened to feed on it. Even on that insect, they did not find any problem. Clear or not? Clear or not? Okay, nucleopolyhedrovirus. Okay, hedrovirus is very insect specific. Okay, and they have a very narrow spectrum. Okay, this is especially desirable when beneficial insects have been conserved to aid in overall integrated pest, that is IPM. Okay, overall integrated pest management program. So students, understand, for everything there is field. That is why I keep telling you, NEET is not the end of the life. Okay, there is actually a lot. Okay, a lot. Okay, students, here also big paragraph. Let's not faint. Okay, just understand, biofertilizers. If biocontrol is there, then biofertilizers also are there. Yes, my tongue is going a little here. Okay. Yes. Yes, okay. Yes, Keith Sanghi is like best student award goes to Keith Sanghi, and YS for giving back answers. Okay, so students, look here. Microbes as biofertilizers. I don't know why am I not able to tell. Biofertilizers. Okay, so what they have to do? Again, they have to improve the quality of the plant, right? Improve the quality of the plant. Okay, water will not help. I think it's just twisting now at this point. Yes, students, this has nothing to do a lot with any names, but just understand, this is basically organic farming. If all of you know, nowadays in market, or even if you open Blinkit, Zepto, all of that, you will see organic spinach, right? Organic apples. All that organic word comes from what? That means they do not use any kind of fertilizers that will harm our body. They use very mild fertilizers, that means your microbes, to actually grow the plant. That is why those are expensive, because biofertilizers are difficult to find, and also yielding quality will be very good, but the time period taken will be more, right? Now everything is what you tell, fast fingers, right? Fast fingers. Yes. Now students, the use of biofertilizers, what is the use? To improve the nutrient content in the plant, right? To improve the biofertilizers are bacteria, fungi or cyanobacteria which are present on the nodules or the roots of the leguminous plant. So rhizobium, All of you remember rhizobium. We have mycorrhiza. So these bacteria fix atmospheric nitrogen. Nostoc. All of you remember Nostoc. Okay. All this. So these people, what do? Atmospheric nitrogen. Nostoc is not an example here, but I'm telling you all the examples of whichever could fix atmospheric nitrogen. So students, what do? They take atmospheric nitrogen, they fix it in the soil, and they will increase the fertility of the soil. That's all. That's all. Fertility. Okay, fertility of the soil. Okay. Now, which is used by plant as a nutrient. Other bacteria can fix atmospheric nitrogen while free living in the soil. Example, azospirillum, azotobacter. Okay. Clear? Only these two examples, please remember, and rhizobium. Okay. I will get this fixed tomorrow, students. This locomotion, it will go. Azobacter. Okay. And azospirillum. Okay. Got it, all of you? Azospirillum. Yes, correct. Azotobacter and azospirillum. These two are the examples. Okay. These two are the example of your free living ones, which will actually improve the nutrients. Okay. Then students, I already told you mycorrhiza. Mycorrhiza again can fix the nitrogen. Okay. Which is many members of genus Glomus. Okay. Glomus form mycorrhiza. The fungal symbiont in these association absorb phosphorus from the soil and passes it to the plant. Very, very important. Genus glomule will take what? Phosphorus. And increase the phosphorus in the soil. Okay, nitrogen is different, phosphorus is different. Okay, yeah, Anabina also is there. Okay, Anabina also is there. Yes, Anabina is there. Azola pinnata is there. Students, many examples are there. But then just understand, remember, like what it is. For as of now, for your this one, only whatever is required, you remember. Extra information for this chapter is actually not required. Okay. Plants having such associations show other benefits such as resistant to root-borne pathogens. Again, we saw the same thing there also. Okay. Tolerance and to salinity. That means salt content. Okay. Salt content and drought and overall increase in plant. So this is what it's going to do. Okay. All this. Improve the plant growth. Okay. Improve the plant growth. Be tolerant. Tolerant to salinity. Okay. Improve the nutrient in the soil. That's all. That's all. Did you get it? Okay. Yes. Yes. Okay. Yes. Now students, last thing. Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments. Okay. Yes. Now which can fix atmospheric nitrogen and Anabina. See here is there students. See Anabina, Nostoc, Oscillatoria. All of this can fix nitrogen. But the usage is actually Nostoc and all usage is actually very less. Okay, usage is a little less. Okay. Okay. Yes. And in paddy fields, cyanobacteria acts as an important biofertilizer. Who acts? Cyanobacteria acts as a very, very important biofertilizer. Blue-green algae also add organic matter to the soil and increase the fertility. Currently in our country, a number of biofertilizers are available commercially in the market and the farmers use. Yes, that's all students. Okay, that's all. Okay, clear or not? So we studied cyanobacteria, we studied Nostoc, Oscillatoria, Anabina, okay, Azola. All these people are there. All these people are going to help for improving the. Oh, I showed the answer. Ready for MCQ? Yeah, heterocyst. Heterocyst is not there in Oscillatoria, but it is there in Nostoc. Okay, nitrogen fixing cells, special cells called heterocyst, but it is not there in Oscillatoria. Okay, very wrong information is given some places. Yes, yes, Preeti, I am teaching for free. Where have I asked you money anywhere? Can you see anywhere I have written please pay this much? It is completely free. Whatever class is happening on YouTube is completely free. Yes, ready? Okay, first question. Microorganisms are used during which stage of purification of sewage water? Microorganisms are used in which stage? Which stage of your STPs? STP. I'm moving from the screen. Which stage? Come on, let's see who's the fastest. Yes, very good. Primary treatment, secondary treatment, none of the above. Both 1 and 2. What is the answer? Secondary treatment, we use something called as flocks. Right? That flocks are actually organic. So, secondary treatment, primary treatment is only about filtration and sedimentation. Very good, very good. Okay? Yes. The maximum concentration of alcohol in beverages that are naturally fermented. Naturally fermented. Very good question. Tell me the percentage of alcohol which are naturally fermented. That means non-distillated. Okay? Non-distillated. Maximum, students, question is maximum, not minimum. Maximum is the question. Which is the answer? 5 to 10, 10 to 15, 20 to 25, 45 to 50. Yes, maximum can be 10 to 15, even though it is 10 to 13. Here they have given maximum, so we'll go with 10 to 15. Minimum, if they would ask, this would be the answer. Okay? Minimum 5 to 10. Okay? Next one. Big holes in Swiss cheese are made by. Okay? The holes in the Swiss cheese is made by which among this? Yes? Very good. Okay? Yes, Tom and Jerry. Okay, Tom and Jerry. Okay? So what is it? Which is the answer? 3. A bacterium producing a large amount of carbon dioxide. Very good. Very good. Okay? Now, a fungus that releases a lot. Hey, fungus in cheese. Huh? That's all, you'll die off. The vitamins whose content increases following the conversion of milk into curd by. Okay? Students, NEET questions only, all these are. They have been asked previously. Yes? Very good, very good. Okay, B12, right? B12, which is the answer? 3 is the answer. Clear? Clear? Yes. Okay, so students, we have completed the chapter. There was some slight disturbances in the middle, but it's okay. So this chapter will have 1 question or 2 questions. That means 4 marks or 8 marks. So I hope we have done justice to our chapter. Okay. Yes. So students, thank you so much for joining. But before you leave, let me tell you, there is NEET treasure box. So please go take it for free. Everything is for free. Okay. Chapter wise session links are there. Session notes are there, because it'll be confusing. Did ma'am teach cockroach and frog? Did ma'am teach molecular basis of inheritance? Did ma'am do that? Did sir do this? To avoid any kinds of doubts, students, now itself below the session you can see NEET 2024 best surprise gift. Okay. Click on that. Okay. Click on that and you will get session links, top teachers, okay, solution PDF, NEET PYQs, chapter wise PYQs, CBSE boards and NEET year wise PYQs. You will get all this for free. Just go and click down. Okay, just go and click down. Okay, clear or not? Click down and take all this for free. Enroll for free. Clear or not? Yes, students, thank you so much. It's not working? How come it's not working? Okay, I will send this on the Telegram. Okay, I'll tell the team. Okay, I will tell the team that it is not working. Yes, students, locomotion on Wednesday. Wednesday. Schedule will happen. Give some time. Schedule will happen. Okay. Oh, all the best, all the best. I know a lot of you will have practical exams. Those who are in boards, right? Practical exam and all this. Okay. This session PDF will be sent on Telegram by tomorrow afternoon. Okay. By tomorrow afternoon, you will get this PDF. Okay. Yes. No. We can't, Sharad, we can't ask things that they're not taught at home. Okay. We cannot ask things that they're not given at home. Yes. Thank you so much, students. Thank you so much. Good night. Sleep well. Take care. Okay, and take care of your health very well, because students, application forms are out. I think most of you started registration also. I have also put a code video. Okay, I've put a code video, so go check which code you have to use correctly, please. Especially the Karnataka board students, do watch that video. Okay, do watch that video. Clear or not? Thank you so much for joining. See you all. Bye. Okay.